Thermal properties of Chitosan-Zinc oxide composite for active food packaging applications.
I am Ubi Umaira, and these are my team members, Akila, Ifa, Sabrina, Alia, Chua, Nurul Huda, Nur Safiza, Amira, and Najwa Umaira.
Food spoilage can be caused by microbial infection, which can be avoided or reduced with the correct packaging. Food packaging's main functions are to preserve, extend the shelf life of, and transport food goods until they are consumed. Chitosan is a biopolymer made mostly from the chitin found in the shells of animals such as crabs, shrimp, and lobsters, which is generally discarded in the food processing industry. Chitosan has a lot of potential as an active material because of its antibacterial properties, non-toxicity, and low oxygen permeability. However, Chitosan alone does not match the criteria for an optimum packaging film due to its high water vapor permeability, which may increase bacterial growth and ineffectiveness against typical bacteria strain that cause food deterioration. As a result, adding an additive to the Chitosan matrix, such as metal oxide, to improve its limited mechanical and antibacterial capabilities is always desirable for improved Chitosan film performance.
For the first objective is to determine whether the Chitosan-Zinc oxide is suitable to replace current food packaging. Second objective is to determine thermal properties of Chitosan-Zinc oxide.
The first method to determine the thermal properties of Chitosan-Zinc oxide composite for active food packaging application is by conducting antibacterial and antifungal properties tests. The antibacterial activity of Chitosan and Chitosan-Zinc oxide composite against gram-positive and gram-negative bacteria was determined using the agar diffusion technique. The medium was made by dissolving approximately 28 grams of nutritional agar in 1 liter of distilled water and autoclaving it for 50 minutes at 121 degrees Celsius. Following that, 40 mm of the medium was placed into a petri dish. A core bore with an 8 mm diameter was used to well the medium, and the bacterial strain were placed with a swab. After that, 2 ml of 0.05 molar of acetic acid was used to dissolve 0.05 gram of Chitosan or complex. Will receive 40 microliter of either Chitosan or complex solution and was incubated for 24 hours at 37 degrees Celsius. The diameter of the inhibitory zone in millimeter was used to evaluate antibacterial activity. Chitosan and Chitosan oxide composite were tested for antifungal efficacy against Aprosycillus fumigatus and Fusarium solani. The medium was made by dissolving 38 grams of molar heated agar in 1 liter of distilled water and autoclaving it for 50 minutes at 121 degrees Celsius. 15 ml of the sodium solution was poured into each test tube, followed by 1 ml of Chitosan or complex solution on the surface of each test tube. After that, the fungus was applied, and the test tube was put in an oven at 27 degrees Celsius for 2 weeks.
After 2 weeks, thermogravimetric analyzer was used to evaluate the thermal stability and degradation behavior of composites. Then, about 10 mg of sample was deposited in aluminum crucible, and the test has been carried out under flowing air environment in 50 to 600 degrees at 20 degrees C over mean rem.
Electrical properties. The process starts with after applying silver conducting paint to form the electrical contacts disposed with an average separation of 1 mm. The current voltage properties of composite were measured at room temperature using Kitley 2400 source meter.
The thermogravimetric analyzer, which is TGA, is the instrument that used to evaluate the heat stability and degradation behavior of Chitosan sample and Chitosan-Zinc oxide sample composite.
Now, for the expected result, the first test is antibacterial and antifungal properties. Table 1 shows the antibacterial activity, which is zone of inhibition in mm of Chitosan and Chitosan with zinc oxide composite using well assay diffusion method. The zone of inhibition is a circular area around the spot of the medium culture in which the bacterial colonies cannot grow. As you can see here, the medium culture in the Chitosan alone already has this zone of inhibition. For example, the Staphylococcus aureus, there is 80 mm of inhibition zone in Chitosan medium. The diameter increased to 24 mm when the culture in the Chitosan with 0.5 gram of zinc oxide. Lastly, the medium of Chitosan with 1 gram of zinc oxide has 30 mm of inhibition zone. Based on table 1, we can see here that inhibitory effect of Chitosan and composite differ with the type of bacteria strain. The low molecular weight of Chitosan might enter the microbial cell more easily through prevention and disturb the metabolism of the cell. It penetrates into the bacterial cell and combine with DNA and inhibit synthesis of mRNA and DNA transcription. The Chitosan might interact with electronegative chemicals in the cell and fluctuate them, its affect the physiological function of the cell and kills them. The antibacterial properties of Chitosan were enhanced with the formation of composites. The composition of zinc oxide with Chitosan further strengthened the positive touch on the amino group of Chitosan, and optimal result were obtained at equal ratio of Chitosan and zinc oxide. Thus, the Chitosan and its complex inhibit a very good antifungal properties.
Thermogravimetric analysis was used to evaluate the heat stability and degradation behavior of Chitosan and Chitosan-Zinc oxide composite. The mass of Chitosan was lost in 2 stages, while the mass loss in the Chitosan and zinc oxide composite occur in 3 stages. In addition, when compared to Chitosan, there are some differences in composite degradation behavior. The degradation rate of the composite was much lower than that of Chitosan, implying that zinc oxide has a good effect on Chitosan's thermal stability. Reduction in the degradation rate and improvement in thermal stability of Chitosan with the inclusion of zinc oxide has been observed. The thermal stability of composite also increased with increases in the zinc oxide content. Figure 2.1 shows the curve of TGA of Chitosan and Chitosan-Zinc oxide composite from 50 degrees Celsius to 550 degrees Celsius in the nitrogen environment, while Figure 2.2 shows the DTG curve of Chitosan and Chitosan-Zinc oxide composite from 50 degrees Celsius to 550 degrees Celsius in the nitrogen environment. The composite has exhibited significantly lower degradation rate than that of Chitosan, which suggests a positive influence of zinc oxide on thermal stability Chitosan. When 50% mass loss is set as a point of comparison, CS2, which is pink line composite, exhibited 143 degrees Celsius higher thermal stability than that of Chitosan. The thermal stability of Chitosan also increased when zinc oxide content. The degradation temperature of CS1, which is a red line at 30% mass loss, is 280.3 degrees Celsius, which is increased by 59 degrees Celsius in CS2 composite.
Electrical properties. So in natural polymers, there are no well-defined band gap. Rather, there are regions filled with trapped states. Chitosan is a conducting biopolymer because of amino groups present in its molecular structure, and zinc oxide is also piezoelectric because of zinc 2+ ions. Electrical properties of Chitosan can be varied with the addition of zinc oxide. However, further increase in concentration of zinc oxide will reduce the mobility of ions, probably because of the hindrance created by it. As we can see here, this is the graph of the expected result for the electrical properties, which is, we can see that here is the ratio between Chitosan and zinc oxide. So as we can see from the graph, as the zinc oxide ratio increase from 0.5 to 1, the electrical properties of the Chitosan will also increase. However, further increase of zinc oxide ratio, as we can see here, which is 1.5 to N2, the electrical properties of the Chitosan will decrease, as I said in the slide before. This is because increase in zinc oxide concentration will create hindrance in the Chitosan.
So this is the example of schematic of the preparation of zinc oxide with Chitosan. As we can see here, this is the packaging of Chitosan and zinc oxide mixture. There are a few precautions should be taken in this experiment. All of the equipment and apparatus such as plate, needles, pipette should be sterilized to prevent contamination. The first one is sterilize equipment and materials. The second precaution should be taken is based on humidity and temperature. Make sure the humidity and temperature was suitable for the chemicals and Chitosan handling. The third one is hazard identification. In this experiment, we use zinc oxide as a chemical. As we know, zinc oxide is very toxic to aquatic life with long-lasting effect. So zinc oxide should be disposed to the container that have an appropriate waste disposal time.
The next precaution is to duplicate the experiment. It can reduce human error and give greater validity to our findings. Other than that, we should calibrate the instrument. TGA should be calibrated to help assure precise and accurate measurements. Lastly, wastewater treatment. Chitosan is very toxic to the aquatic organism, thus never allow wastewater from cleaning or processed equipment to enter drains.
So we can conclude that Chitosan packaging including zinc oxide will enhance the perform better than Chitosan packaging alone because zinc oxide improves the thermal stability of the packaging. It is possible to infer that the biocompatible, environmental kindly and low-cost Chitosan zinc oxide composite can be employed for the food packaging and biomedical applications.